Welcome to my channel Adhyan with Flow. Let's start with our new topic is plasticity. We can also call it as a phenotypic plasticity because why I call phenotypic because they change their outer appearance. What is the meaning of plasticity? Plasticity means assume you know about plastic. They change their size, they are very elastic in nature, you can modify it easily. Okay, now let's start. Plant follow different pathway in the response of environment or phases or of life to form different kinds of structure. This ability is called it as a plasticity. According to environment, guys, according to environment, plant plant adapt different feature. Why they adapt? For survival for survival. This is called it as a this ability we can call it as a plasticity. Now let's take example of plasticity. Heterophile in cotton, or it may be coriander and it may be larkspur. Here, this is the example of larkspur. At a juvenile stage, they appear different, their leaf is different, but in their adult phase they change their outer appearance, leaf size. Look at here now, juvenile stage and adult stage, there is a huge difference. This is the same plant, both are larkspur. This is the childhood picture and this is the adult picture. Now, in such plant, the leaves of the juvenile plants are different in shape and size from those in mature plant. This phase totally different from their mature phase. Let's take another example of buttercup. Look at here. This is the buttercup. This species present in terrestrial and this species present in water habitat. Look at there. According to their environment, they change their structure. They survive in terrestrial environment, so they increase their leaf size for photosynthesis, for gaseous exchanges, for H2 exchanges, transpiration. Here they already present in water. Look at here, this is the structure of water buttercup and this is the structure of terrestrial buttercup. Due to effect of environment, they can change their outer appearance. And this is the one of the example. They are automatically adapted by birth. This feature is automatically adapted by birth. And these features, they adapt according to their environment. So this is plasticity's example. Clear, guys? Now let's start. Guys. Plant growth regulator. I previously said that only meristematic tissues are not responsible for plant growth. This is not only one factor who contribute in plant growth. They are a huge factor, like oxygen, like CO2, like nutrients, minerals. Alright, guys, water, temperature, light also. These are the factors who affect the plant growth. And now next thing is some regulators, some growth regulators. We can call it as a hormone, plant hormone. Clear, guys? Now it's clear. So these are factors which contribute growth of the plant. Let's start with some introduction. In your NCERT, here they give some hormones name and little bit description. First, like auxin. Next, zebralic acid. I'll explain. Next, ethylene. Next, cytokinin, and your ABA, abscisic acid. We can also call it as a stress hormone. First, we have to discuss their scientist name. What? Scientist name. Who discovered? Let's start one by one. Here, look at here. We are starting with our auxin. This is the phytohormone. We can also say it is a plant hormone. The name of the scientist is mainly Darwin. They discovered, they don't give their name, they discovered. Actually, auxin was isolated by F.W. Wendt. Okay, but in previous, they contribute their some prospectus towards auxin hormone. What they found? What they found? They take what? They take canary gases. They take canary gases. Now, what they think? They saw that canary gases growing towards the light source. Yeah, means they show phototropism. Means the canary gases move towards the light. Okay, this term is clear. After a series of experiments, it was concluded that the tip of collectile was the site of transmittable influence that caused the bending of the entire collectile. Means here not explanation, but I will explain you. What they do? They take what? They take canary grasses tip, collectile part. Okay. Now what they see? That this canary gases move towards the light source. Moves towards the light source. Then what they do? They cut this cap. They want to know that what is the molecule present here. They bend completely collectile towards the light. So they cut the cap, remove the cap, remove. Now they still watch. After cutting the cap, after cutting the collectile part does not bend. After cutting the collectile part, this part, after cutting this part, what happened? This collectile does not bend. And then again they attach this cutting piece. Again they attach this cutting piece. They saw, yes, again this collectile canary grasses tip responds toward the light. Means in a first step, they take canary grasses collectile. With the exposure of light, with the exposure of light, what they watch? This part bends toward the light because light source located here. Now what they do? They cut this part, remove, and again exposed with light. But they saw there is no bending. Then they calculate, then they conclude that something is in their tip. Something there is in tip. So they can bend towards the light. Now what they do again? Again they join this cutting tip. They join this cutted part. And they watched, and they again exposed with light source, and they still watch that yes, this part again bends toward the light. So they don't know what is the content present here, but they discovered something is present in here. Now then FW went, they discovered, they isolate, they remove this cap and isolate in their lab. And what kind of thing is present here? Then they found auxin. So this is the discovery of auxin. Now next discovery of gibberellic acid, the Bicane foolish seedling disease. Here discovery of gibberellic acid is done by their disease. They found that rice plant have medium height. Rice plant have medium height, but somehow the infection with some fungal infection with some fungal, what they watch? The plant height will increase suddenly. Tall, very tall. And the farmers are happy. They got this is fungal. This is very important. They increase the plant height. Maximum food. But no, they are wrong. This is the diseased plant. So according to their disease, they found then which kind of thing is promote the stem elongation. Which kind of factor promote the stem elongation? Means if some plant affected with fungal infection, then that kind of plant have to die, but they're still alive and long in size. So what kind of factor fungal increase in plant? So they found gibberellic acid. Who discovered this thing? Kurosawa. This scientist discovered this thing. They found rice plant affected with some fungal pathogen, Gibberella fujikuroi. So this plant affected by Gibberella fujikuroi and infection with Gibberella fujikuroi, the plant height increase. So this hormone discovered by their disease. The appearance of symptoms of the disease in rice seedling when they were treated with sterile filtrate of the fungus. The active substances were later identified as a gibberellic acid. Later, they discover which kind of factor promote height. Then they found gibberellic acid. Now, another discovery related with cytokinin plant hormone. how this hormone is discovered. F. Cook and their co-workers observed what? That from the internodal segment of tobacco stems, the callus proliferate, means they take callus. What is callus? This is a mass of undifferentiated cell. Undifferentiated means they divide, but they are not specified right now. In addition to oxygen, the nutrients medium was supplemented with one of the following extract of vascular tissue: yeast extract, coconut milk, or DNA. Means what they found? They take callus from stem. They take some internodal part of stem and they take in the lab and filled with. They put this callus into the nutrients medium. In this medium, they contain this thing: some yeast extract, some coconut milk, and some DNA, and medium amount of oxygen. What they watch, what they see, that callus, callus will grow and differentiated into shoot and root. Means cytokinin is responsible for cell division, cell proliferation. So next phytohormone is our abscisic acid, ABA. During 1960, three independent researchers reported the purification and chemical characterization of three different kinds of inhibitor: inhibitor B, abscission second, and dormin. Remember this name. They all are named as a abscisic acid because they inhibit, because ABA inhibit cell growth. They inhibit cell proliferation. They inhibit cell division. They inhibit seed germination. They inhibit flowering. So they found some inhibitors. The name call it as a abscisic acid. It is also called as a stress hormone. They inhibit. Why they inhibit? There is multiple use. If ABA not present in the plant, then seed can germinate before their timing. Cell can divide and divide and divide, grow without any permission. Flowering, maturation stage come first. Okay, synesis is fast with the absence of ABA. So this is the beneficial hormone. Now next hormone is ethylene. HH cousins 90 confirm that the release of a volatile means they can flow in the air from ripened, means mature fruit. What they do? Ripening of store unripe bananas. What they do? They take, wait a minute, they take orange. They take ripened, mature orange, and this mature orange they put into the unripe bananas here, and after some time they watch all bananas ripen. So there are some volatile substance who ripened the food. The name goes to ethylene. So there is some scientist introduction about the plant hormone. In our next video, we will discuss one by one our hormones. So let's meet in our next video. That's all for now. Thank you for supporting me, and please subscribe my channel. Thank you so much. Keep smiling. Thank you.